All right. So we have chapter 12 today, and chapter 12 looks at basically the central dogma of biology. And the central dogma of biology is transcription, translation, protein. Okay. Transcription is going to happen in a specific place. It involves some specific things. Translation occurs in a specific place, and we have some specific players involved with that. It would make a good short answer question to discuss how this central dogma of biology happens, what it means, all that fabulous nonsense. But we're going to look at a little bit of replication, and we talked about replication. It's in the S phase of interphase. So we're going to expound a little bit more upon that, just enough to maybe peek behind the curtain a little bit to kind of see actually how it does the replicating of DNA. Whoops. We'll look at what is the genetic material. We obviously know what in the world it is, but we'll do a little bit of history and kind of see where we started and how we ended up here. We didn't really know it was DNA that was this genetic material, and we hinted at some of that when we were discussing Gregor Mendel and his discoveries, and then, other than Mendelian genetics or inheritance patterns, because a lot of things don't abide by Gregor Mendel's laws that he came up with. A lot of things don't. So we hinted at some of that. So we'll look at some of the history here after Mendel. Replication. We'll look at the genetic code of life. Okay, what all those A's and T's and G's and C's and U's all mean. And then we'll probably get through that today and then finish up with transcription and translation on Wednesday. So here, I'm not going to ask any short answer questions involving some of the history of it all, but they will be seen as either multiple choice questions, maybe a matching question. I haven't figured out how I'm going to ask them yet. But it all starts here, kind of trying to figure out what's the genetic information. What is the stuff that's passing on this ability to adapt and learn, to get information to pass on to one generation to the next so it can survive. And it starts here with Frederick Griffith. He was looking at how strong, that's what virulence means, strep pneumoniae is, and streptococcus. Coccus is just circular beads of this specific strain of bacteria here. And it's kind of interesting here. I believe I have some pictures of how they looked at, some images of how they saw and how they witnessed ultimately, was it DNA or was it protein, or what was specifically kind of being transferred, passed down from one generation line to the next generation line. And so Frederick Griffith, he investigated pneumonia, admit, and ton of work with strep. And they were able to see that they could pass the strong virulence of strep pneumoniae from just a dead strain to a non-virulent living strain. So they were able to take this basically dead bacteria, okay, and they were able to get that dead bacteria, give the virulence inside that guy into a non-virulent form of it. And they refer to it as a transformation, okay, because they were able to transform this non-virulent strain into a virulent strain, okay, something that didn't cause any harm to stuff that caused a lot to the host. Avery took Griffith's work. So Avery and a number of others, et al. Means and others. They discovered that what was being passed on was DNA. It was this transformative substance. And it was actually the DNA from these dead cells that was being incorporated into these, live non-virulent strains. And this was pretty interesting because everybody at the time just assumed it was proteins that got passed on from one generation to the next. And we didn't truly understand the concept of DNA and whatnot. So this whole, is it DNA or is it proteins? There were camps for both. And which seems kind of weird and bizarre now because, it's pretty common knowledge that it's just DNA that's being injected into the host cell, and then it incorporates that into the host DNA, and then the host cell produces all the crap for the bacteria, for the virus. So it seems pretty obvious to us now, but back then they didn't really understand and believe and think about all that. So we had 20 different amino acids that could be sequenced in a number of different ways. So because we had so many amino acids, they're like, man, it's got to be proteins because we have so much of it. They're like, we only have 4 types of nucleides with DNA and RNA, and 4 compared to 20. So it had to be, for the people that thought it was proteins, there's so much more there that they can combine in a number of ways that is a lot more greater than just 4 nucleotides, that it had to be these dudes. But for something to be passed on, we have to be able to store it, okay? We have to be able to store it. And when it comes to the human body, we don't have a lot of abilities to store excess protein. Protein is really for structure, for information, and the body utilizes proteins. As soon as we make a protein, it gets utilized. So you can't really store protein per se. The body just assimilates it into stuff, into structure. So we have this requirement for the genetic material. So really that's 1x against proteins, right? Okay. We have to be able to replicate these things accurately. And surprisingly, the copying of DNA, that synthesis of DNA that occurs in interphase, it happens at a pretty good clip in terms of accuracy. The DNA does a great job of, if it does screw up, it will actually stop itself and try to go in there and fix it. So it does a good job of reading and catching itself in case there is a missubstitution or misread in the DNA. Excuse me. So it's really fairly accurate in that term. And it has to be able to undergo mutations. And I know we haven't really talked about this. We mentioned it with meiosis. It's that variability of independent assortment and crossing over that gives a huge amount of genetic variation that we discussed. But there's also this thing that's called mutations. And mutations we'll talk a heck of a lot more about in the next unit in evolution, because mutations are what help to drive evolution. They give this thing called variability. And for some mutations, they don't do anything. Okay? At least we don't believe they do anything. But it creates a variability. And when I say it doesn't necessarily create anything, it doesn't create anything bad. Like, this is so detrimental that this individual who has this mutation is at risk of dying or anything like that. If it's that kind of mutation, then obviously it has no damn advantage. But there are a number of mutations that convey no significant loss of function, but they give a little bit of variability. And if something happens in the environment, we have this potential mutation that can give this potential fitness, this ability to survive and advance, right? And then that variability, that mutation becomes the norm for everybody then. So this is going to be how speciation happens, how small little changes in the genome can happen at one point and then continue and take a animal, and then all of a sudden it branches off. And this is going to be the basis for evolutionary things. So this is kind of a step into that next unit that we're going to talk about. So doing all of this research, they were able to prove that the DNA, and it was not the proteins, were this creature behind the curtains that was actually passing on this information, okay? Passing on this information from one generation to the next, because it's the only thing that fits all of those capabilities, right? Because when we talk about proteins, if we, for a lot of them, if you just change one, it changes it to a totally different protein and you get a totally different function. So when it comes to undergoing mutations to provide the same thing, proteins don't necessarily do that. And when we look at the genetic code of life, we'll see some of that variability that's there when we just change a couple of them. I'm not going to ask you to know that sucker. Maybe one thing from it. But this DNA material does do all of these requirements. It's able to store, it's able to replicate pretty dang accurately, and it's able to undergo mutations and keep its variability. So these are what DNA can do. These three things were really as to, yes, DNA is the key to passing down information from one generation to the next and not proteins. So they're like, prove it, right? Prove it. Show me the money. So then these dudes, Avery, McLeod, and McCarty, they looked at all of this information. And then they're like, okay, so let's figure it out. How can we prove it's DNA and not proteins that are passing down this information from one generation to the next, okay? So this is how they proved it right here, all right? They used enzymes, okay? They used enzymes. They used a DNase, okay? This is an enzyme, okay? Whenever you see ASE on the end, it's always an enzyme. And DNase chews up DNA, breaks down DNA. Sorry. They used another enzyme, RNase. And then they used another enzyme called proteases. And they used these in individual experimentations, okay? Again, Streptococcus was used, okay? The only enzyme, okay, that had an effect was DNase, okay? When they treated strep with the protease, it destroyed the proteins on the surface. The markers that say, hi, it's me, strep. Nothing happened. There was no transformative factor, okay? That was prevented. Same thing for RNA. Did the same thing for their RNA, okay? The only one that had any effect at all was that DNase. That is the only one that prevented this transformation from occurring. So all of these experiments, this is how it proved it was DNA. Experiment saying it's DNA. Another experiment saying it's DNA. Another experiment saying it's DNA. Scientists, man. I want to see it. Show it to me. I want to see it. Okay. So then Hershey and Chase like, okay, dude, I got you, punk A. They tagged it. Okay. Put a radioactive isotope on the DNA. And then they were able to watch. Okay. They were able to watch. And then that's what you're seeing right here. You're seeing this thing kind of get, pooped into the bacteria. Okay. So here's the virus, and here's the host cell. And here you can see it's all lit up like Christmas. And here you can see all that DNA is getting, all this is DNA inside these things. And here you can see it's all getting deposited here on the inside. Okay. During this experiment, it's pretty interesting. They tag these DNAs, and then they watched it kind of go in, and then they treated this bacterial cell. I can't remember what they treated it with, but then you agitate it, and then you kick off all those things that are attached, all those viruses that are attached on the outside. You get rid of them. And then the bacteria is able to divide and reproduce, through mitotic type means, right? Through binary fission. And then they were able to see that transformative factor, DNA, stayed within the bacteria. It was inside the bacteria. It was never protein. So once you got the visualization of it, they're like, cool, I believe you. And this took, I don't know, I think maybe 20, 30 years of kind of, 50s was when Watson and Crick did their thing. This is probably around in the 30s and 40s. 40s, I think. I can't remember off the top of my head. I don't ask dates because it doesn't really matter, to be honest. It's not a history class. But, 10 years of kind of going back and forth. Is it DNA? Is it proteins? Is it DNA? Is it proteins? And you can see the scientific process. It takes a long time to figure it out. That's what science is. There's 3 studies here where it showed that, it might be DNA. We think it's DNA. And then the next 2 basically said, yes, it's DNA and not proteins. And then we have another study here that they had to do because we had to see it. Science requires testing and testing and testing and testing. This took 10 years. We're trying to figure out COVID on the fly before an election. Get the hell out of here. Okay. Why is there flip-flopping of scientists? Why is there always new information coming out? Because we're understanding and learning a whole new strain of this COVID creature. We've had other COVID creatures in the past. We're learning this one. And as the general population who doesn't do science on the daily, doesn't understand it, stuff changes. You get more information, you get a better idea of the picture. I was explaining to my oldest, he's 12, turns 13. He's doing science. He's doing what we do in the very beginning, looking at a microscope and all that stuff. And, I'm trying to explain to him this process of, do you start looking at, on 4x or 40x? And, if you start on 40x and you're right here, if I'm trying to look at my hand here, I can't really see a whole hell of a lot of detail about the hand. I can see that one spot. But if I pull back, I get a great understanding, a greater view of my hand, and I get an understanding of what this thing looks like. And with data and with an experimentation, you get more and more information, and that helped to make this image of a hand much more obvious if I can do more and more research on it. What can it do? What can't it do? And when we're trying to figure out what COVID's doing, we know it attacks specific receptors within the lungs. But there are a number of things that we don't understand about it. How it's susceptible. How is it so virulent? Why is it so virulent with certain people, with certain ethnic races, right? COVID, African-American descent individuals, good God almighty. It attacks them at a much greater clip than someone who's not of African-American descent. Those that have diabetes, those who are obese, you have a higher rate of failure death. Why? Well, you're in a pro-inflammatory state. What does that have to do, though? Does it attack the inflammatory pathways? Yes and no. It affects some of them. What are inflammatory pathways? Have you been under stress at all in your life? I'm going to assume it's yes. You've experienced the inflammatory pathway. Diet plays into that. What role does diet play into it? Everybody is a Molotov cocktail walking around. What you put in your body is different from this person over here. What's different from this person over here? Different from grandma who takes 13 to 15 different damn medications. How is it going to treat her? I don't know. She's a Molotov cocktail. I don't know how the bug is going to treat her any different from me or an 8-year-old or a 3-year-old. And so it takes time to follow this over the course of the bug's lifetime. It takes many experiments to understand the variations that can happen in somebody in their 20s compared to somebody in their 30s, compared to somebody in their 40s, a guy, a girl. And then you split that up, you start splitting hairs on medication, on no medication, on 2 medications. What are the comorbidities? Diabetes, high blood pressure, asthma, lung issues. There are a number of different things that go into it. All of this takes a lot of time. And you see that here in all of these experiments over 20 years to try to figure out what the heck this stuff is. And it all started with Gregor Mendel doing it in the late 1800s. And then here we're almost a century later and we finally figure it out what in the heck it is. It's crazy. Science is crazy. Science takes a long time. And if you ever kind of are out there looking at different things, reading different research articles that come out, hell, even in nutrition, right? So much changes all the time. And we just get a much